Welcome to part 6 of week 7 of the class Neuronal Dynamics. Now we have all the tools ready. Parameter optimization of neuron models. Let's now look at some data, and we focus on in vitro data. So our ideal neuron model takes some current input, and our aim is to predict voltage and to predict spike times. And we do this using a GLM formulated as a spike response model with escape noise. Since the voltage is linear in the parameters, given the input current, given the observed spike trains, since the threshold is linear in the parameters, given the observed spike times, since the firing intensity is a convex function of u minus theta, we have the powerful tools of GLMs at hand to optimize these parameters. And the parameters are the shapes of the filter kappa, filter eta, filter theta 1. And this is, after parameter optimization, how it looks like. We have the neuron, the real data, with a thin line, and the model with a thick line. And you see that the subthreshold voltage is very nicely represented. Most spikes are perfectly predicted on new data, data that was not used for parameter optimization. There are a few missed spikes, there are a few extra spikes. If we repeat the same input current, the same realization of all this fluctuating current several times, then the real neuron will emit spikes with a slight jitter between one trial and the next. And so does the model. You may wonder why we need the moving threshold. Well, in fact, for an inhibitory neuron, you don't really need it. For an excitatory neuron, the percentage of correctly predicted spikes, spikes that are predictable, that are reliable, is much better with the moving threshold than without the moving threshold. Note that the filter eta could also be used to optimize this quality factor m of spike times. However, then we would lose the possibility to describe the subthreshold membrane potential. So we need to separate eta for the subthreshold membrane potential and have theta 1 for optimizing the spike time prediction. Now there are two different model variants. At the moment, I've discussed the situation where eta, the spike after effects, were described as a potential and fed in here. We now feed eta in here before the convolution kernel for the current. So eta acts like a current. It's now an adaptation current. Moreover, we have seen on the previous slides, the data slides, that the filter kappa is exponential. Now, if you look at the differential equation and we integrate some current I of t, then this gives effectively a convolution filter kappa that is exponential. In other words, the voltage equation can be rewritten as a differential equation. So this filter eta tilde for the adaptation current is inserted into the differential equation for the membrane voltage. The threshold equation and the firing intensity equations don't change. So what are the units now? Well, I have here units of current. This is a current. So the total thing should be also a current. ds means integration. S is a sequence of delta pulses. If I integrate out the time, they disappear, so eta has units of current. It's really an adaptation current. Now this formulation with the differential equation can be called a soft threshold because I have this escape rate function, adaptive because I have theta 1 and eta, integrating fire. It's integrating fire because I have this differential equation model. Now if you look at the shape of this filter as a function of time, this is seconds, not milliseconds. This is really seconds. Then this filter is extremely sharp, goes down rapidly. Now if you zoom in, you would find that it's not zero here. On a log-log plot, this is what it looks like, and it's basically a straight line. And that means it's a power law. Similarly, this function of the dynamic threshold, theta 1, looks very sharp. But if you plot it on a log-log scale, then you have this linear function. Again, it's a power law. Now, these are again units of seconds. 10 to the 0 means 1 second. 10 to the 1 means 10 seconds. And you see that the power law extends to at least 10 seconds. Therefore, if you answer the question, how long lasts the effect of a single spike, the spike after effect, the spike duration itself is just 1 millisecond or 2 or 3. But a single spike has an effect still 10 seconds later. When I first heard about this result, I was really surprised. So these adaptation effects, they are not just 50 milliseconds or 200 milliseconds. No, they extend over several seconds. They are strongest for short times, for 10 milliseconds, for 50 milliseconds. But you still have a measurable effect 10 seconds later from a single spike. So we can extract parameters for in vitro data. We find a dynamic threshold that's distinct from the spike after effects. The spike after effects can be identified as spike after currents, currents that are triggered in the neuron at the moment of spike firing. Now all this is modeling of in vitro data. An experimentalist injects a current into a neuron and measures the response. Even more interesting maybe is the application to in vivo data. And that's coming next.